Thank you for joining us today. I'm Francesca, and I work in the Obs and Gynae department at the Addenbrooks, and I will be chairing today's event, which is part of the Early Researcher Seminar Series, hosted by the Cambridge Reproduction SRI. I would like to say a few things before we start. First of all, we would like to thank the volunteer coordinators for organizing this series, to all the speakers, and also Christina, the SRI coordinator who put all this together and makes the events run smoothly. As you all know, the Cambridge Reproduction SRI is an initiative that brings together researchers from across the university who have an interest in any aspect of reproduction. It's easy to join, so the membership is open to all staff and postgraduate students. And you can just download the membership form in the SRI website and email the form to Christina. Christina maybe can put the link in the chat for everybody. So this is the last seminar of the academic year, but they will start again in the autumn. So postgraduate students and postdocs who would like to participate, please just contact Christina and let her know. The seminars are usually held online on the second Thursday of each month at 1 p.m. And the details of the coming seminars will be on the SRI website. So we have three presentations today, so we better get started. Each of them will be 15 minutes and we'll have question and answers at the end. So now, please mute your microphone, and then at the end, if you have a question, just put it in the chat or unmute the microphone and just ask the speakers. We'll go to our next speaker, Megan Sheridan. She's a postdoctoral fellow in pathology. She works with Ashley Moffett and Margherita Turco. The title of her presentation is Characterization of in vitro human trophoblast models, Advantages and Limitations. Thank you, Megan.
Hi, everyone. I'm Megan Sheridan, and today I want to tell you a little bit about the characterization of in vitro models of human trophoblast. So the placenta is a multifunctional organ, and it's really important for the baby's development while in utero. And you can see the placenta forms this barrier between the uterine wall of the maternal uterus, and it functions as many different organs for the baby as the baby's developing. So it acts as the lungs, the liver, the heart, all of these things for the baby while the baby is still developing. And the reason that we're so interested in early pregnancy and studying this is because early on, when the embryo attaches into the maternal wall and starts to develop, many of the disorders that we see later in pregnancy arise from problems during this time point. So we really want to know how placenta development is regulated and what could potentially go wrong during this early time point that could cause some of these developmental disorders like preeclampsia, preterm labor, stillbirth, all of these things. And what you can see here is the structure of the human placenta. And you get these branching villous trees that invade into the maternal wall and establish blood supply between the mother and the baby. But there are many challenges to studying placenta development in humans, mainly ethical considerations, access to tissue, things like this. And there's also many anatomical and timing differences with the mouse. And until recently, a lack of relevant in vitro models. However, in 2018, two separate models were developed and established. So trophoblast organoids, which were developed by Margherita Turco, and they are three-dimensional structures that represent the placenta. And then there was another two-dimensional model, so trophoblast stem cells developed by Okai and colleagues. and these can differentiate into the different cell types of the placenta. And so the aim of my project was really to use these models and see how we can study the maternal-fetal interface during pregnancy, and also how well do these models represent the in vivo tissue, so how it would look really during normal pregnancy. So here is a diagram of the placenta, and what you can see is the villous tree-like structure is shown here, where you have a villous cytotrophoblast lining the placenta, and then a multinucleated syncytial barrier that's going to basically act as a transport and exchange between the intervillous space. So the intervillous space is going to be filled with maternal blood. So this is directly the communication between the mother and the fetus during pregnancy. And then you also have trophoblast cells that will invade into the uterine wall and migrate and remodel the spiral artery to open up blood supply. And they'll also interact with maternal cells in the uterine wall. And so the trophoblast stem cell model represents the villus-like stem cells found in the chorionic villi, and they can be derived from first trimester tissue or collected from outgrowths of blastocysts. And then in culture, you can differentiate them either into syncytial trophoblasts, so these multinucleated cells, or extravillous trophoblasts, which are the cells that migrate and invade into the maternal wall. And similarly, the trophoblast organoids, which can also be derived from first trimester tissue, represent the villous and syncytial-like portion of the placenta, and then also can be induced to differentiate into these invasive extravillous trophoblast cells. But one major characteristic of trophoblasts is really this unique HLA expression. So what is HLA? HLA is the human leukocyte antigens that are cell surface proteins responsible for the regulation of the immune system. And so there are 6 different alleles. The major alleles are HLA-A, B, and C, and these are highly polymorphic. And what you see in transplants or cancers is that these highly polymorphic cells will actually induce a non-self response when recognized by your immune cell in your body. And then there are minor alleles, HLA-E, F, and G, that are more monomorphic in nature. So these will be less heterogeneous between basically different people. And HLA-A, B, and C are expressed in most cell types found in the body. However, the placenta has a really unique combination of HLA molecules. Firstly, the non-trophoblast cells, these will be the fibroblast mesenchymal cells in the core of the placenta, have high expression of HLA-A, B, and C. However, these are never in direct contact with maternal cells because they have this barrier of trophoblast in all cases. So even though they'll have different HLAs than the mother, they never actually see the mother's immune cells. And then the villous and syncytial trophoblasts that will be in contact with maternal blood actually have no HLA present at all. So this sort of allows them to remain invisible to the maternal immune system. And then further, the extravillous trophoblasts have a unique set of HLAs. They only express HLA-C, E, and G. And because G is so monomorphic in nature, this might allow the natural killer cells to positively interact with the extravillous trophoblast and not cause this sort of non-self normal immune response. And just for visual effect, here's a section of the human placenta, and you can see in pink the trophoblast cells that are invading into the maternal wall, interacting very closely with these maternal natural killer cells. And so what this really indicates is that this is a positive relationship between the maternal and fetal derived cells, and not a normal scenario where these immune cells would actually attack a foreign cell. And you can look at cross sections of the placenta and stain with various HLA antibodies to see this basically this pattern. And the W632 is a general marker which will recognize all class 1 molecules, whereas G233 is very specific for HLA-G only. And what you can see nicely here is that the core of the villi in the human placenta stain positively for any class 1 marker, but are completely absent for HLA-G. And then if you look at in these anchoring columns where these extravillous trophoblasts would be invading into the maternal wall, you can see that they stain for both the pan class 1 and HLA-G. And so this is a pattern that's very well described in the literature. And we can also use control cell lines like JEG and JAR, which are choriocarcinoma cell lines, to look at this HLA pattern as well. So the pan class 1 expression will show up in JEG3 cells, which are most like extravillous trophoblast cells because they're double positive, like the extravillous trophoblast would be. However, JAR cells are most like villous trophoblast cells, and they are completely null for any HLA molecules. So they're absent of any of these molecules on their surface. And then a non-trophoblast cell line shows pan class 1 expression, but is absent of HLA-G, meaning that it's most like these stromal non-trophoblast core cells. And if you look at, sorry, I can't get out of here. So just to show the controls here, you're looking at the isotype control in orange for these class 1 markers, and the JAR cells show up in green as negative for both class 1. And then here again, the JEG cells show up as double positive. And when you look at the trophoblast organoids, they are negative for all class 1 as well. And so this is really nice because that's what you should have in villous-like trophoblast. However, when we looked at the 2-dimensional trophoblast stem cell model, you can see that the class 1 is positive here, and they are negative for HLA-G, which is actually most like this non-trophoblast cell line here. So we were a little confused by what this might mean. And just to show for background as well, the trophoblast organoids do upregulate HLA-G, but only when induced to differentiate to extravillous trophoblast. So you see that the trophoblast organoids really work in between these two properties of JAR cells, which are like villous-like cells, and then JEG3 cells, which are like extravillous-like cells. So then we asked, what exactly do these TSCs express? Because if they have this class 1 expression, but not HLA-G, they're kind of stuck in between this transition. So we wanted to know if they expressed HLA-A or B at all, because no trophoblast cell would ever express HLA-A or B, as that would elicit an immune response uncharacteristic of normal pregnancy. And we had these cell lines typed so that we could know the specific alleles present so we could choose our antibodies appropriately. And what you see here is these TSCs do express levels of HLA-A and B. And further, when they're differentiated to extravillous trophoblast, this HLA-A and B expression remains. And you can see that they do upregulate HLA-G like they should when differentiated to EVT. However, the problem with this is that if you have those migrating extravillous trophoblasts into the maternal wall expressing HLA-A and B, this will interact negatively with natural killer cells or could potentially cause problems. And then we also check this in the trophoblast organoids. So just to check that when they were differentiated to EVT, because they do upregulate class 1, as shown by their HLA-G expression, they don't upregulate HLA-B. So this was just kind of proof of concept to make sure that they still suppress the expression of the non-trophoblast-like HLAs. So then we started to think maybe if we put these trophoblast stem cells into the same exact conditions as the trophoblast organoids, that might be affecting HLA expression. So we took the 2D cultures and placed them in Matrigel droplets, and we cultured them in the organoid media instead. And what you can see is that when you culture them in 2D versus 3D, you actually get a downregulation of HLA expression. And so this is starting to suppress the expression level of this odd HLA. And this can be quantified here. So you can see the decrease in all of the various, these are all different donor cell lines. And to conclude from that, the trophoblast organoids do recapitulate HLA expression patterns seen in vivo. And so this is really important because we can now really start to address maternal-fetal interactions in vitro. So we can actually put these cells together and co-culture them and be confident that they're going to recapitulate what would be happening in pregnancy. However, the TSC do express these classical HLA-A and B molecules, which are not seen in trophoblasts in vivo. So it's important to recognize that if you put these in a co-culture with natural killer cells, that the effect that you get may not be representative of pregnancy. And that also altering the microenvironment really helps reduce HLA expression, yet the mechanism is still unclear. And very little is known about how HLA is regulated in trophoblasts because it's so unique to any other cell type in the body. And so this opens up a lot of questions and hopefully will provide models that we can really study and figure out the mechanism of HLA regulation in trophoblast in general. And with that, I'd like to thank my supervisors, Margarita Turco and Ashley Moffitt, and then all of the members in the lab that have really helped me with this work, and the CTR Bioinformatics, Department of Pathology facilities, our other collaborators at University of Cambridge, the nurse and donor patients from NHS Cambridge Hospital, and also Steve Marsh for the HLA typing for this project, and then of course our funders.